are very important pests, and we do different sorts of experiments. We do the experiments in controlled environment chambers, and you can do experiments, as you see here on the right, in clip cages. I don't know whether you can see my arrow, but aphids are put into clip cages and you can measure immediate responses in the leaves. Alternatively, you can place one aphid on each plant and let that aphid reproduce for a period of days, usually 14, and then you can measure the fecundity and the reproduction of the aphids. We work on quite a range of aphids. Here is an example of a project that I have in collaboration with a colleague, Anna Maria Botha here, who works in the University of Stellenbosch in South Africa. And she works on the Russian wheat aphid, which is a specific aphid for cereals. As you can see there, it was introduced in 1978, and since then it's become a very prevalent pest, causing significant damage on cereals such as wheat and barley, which are only thought to be on the increase due to climate change, and also the appearance of new biotypes. So since 1978, there have been an evolution of the aphids to produce different, more resistant biotypes. This slide, I think, emphasizes the problem with pests. I just picked one of the papers that are in the literature discussing how insect pests are thought to going to increase as a result of climate change in the future. And I've just picked out several points here. So, at the moment, about 1/3 of the crops in the world are lost to insects and pathogens. And this is thought to only going to increase in the future. There are the climate change impacts associated with abiotic stresses, such as a warmer, drier world. But on top of that, modeling suggests that insects will spread much more rapidly. And the conclusion of this paper here is that yield losses will increase by 10 to 25% per degree Celsius of warming. And the effect will be hardest in the temperate zone. And so this is a warning that we have to be very careful about future changes in insect pest populations and the distribution of insect pest populations. So one of the insects of the aphids that I work on most of the time is Mysis persicae, the green peach aphid. It's widely distributed over the world, and it has a broad host range. It's a generalist feeder. It does damage due to its feeding, but like most aphids, one of the most important problems is caused by the transmission of viruses. So when an aphid attacks a plant as any insect, you have local responses which are due to the feeding of the insect. But in terms of the plant, it's important to remember there are rapid systemic responses. So within minutes to hours, there are signals that are sent from the infested leaf to all of the other parts of the plant that are involved in protection. And I just wanted to show you one type of experiment which demonstrates the importance of this systemic effect. So if this is an Arabidopsis plant, you add aphids to one leaf and thereafter look at the survival of nymphs once you remove the original infestation. So you can look at the presence of nymphs on the leaves that were infested and the presence or survival of nymphs on the systemic leaves, which have never seen the aphids themselves, but they're on the same plants. And you could see that the nymphs do not do well on the systemic leaves that have never seen before the aphid infestation. So systemic signaling is vitally important in plant responses to aphids. So like many teams in the world, one of the first things that we did was take a transcriptomic approach to understanding the responses of the plants to aphids. If you look at the literature in terms of aphid responses, the results are very variable. And that's largely because of differences in the experimental design. And even within the context of maize persicki and Arabidopsis, there are wide variations in the data that have been achieved. But often, as expected, the responses highlight the key roles of phytohormones in the plant response to aphids, particularly salicylic acid, jasmonic acid, and ethylene. But actually, when we started, nobody had really looked at the systemic transcriptome changes. So this is how the leaves that have never been infested respond to the presence of aphids on the infested leaves. So our basic experiment was to take transcript and metabolite profiles over a period of 48 hours. So adding clipped cages with 60 aphids at different time points. So 48 hours, 20, these are separate leaves, separate plants, 24 hours, 6 hours. And then measuring the transcript and metabolite profiles thereafter. And you can look at the responses within the leaves. So this is the transcriptome response, the number of transcripts that are changed. This is the increase. So within 6 hours, you see there are increases, but there is a much more or a larger response at 24 hours. On 48 hours, it starts to go again. The effect on less abundant transcripts that are less abundant is much smaller. But when you look at the systemic leaves, the response at 6 hours is about the same magnitude as in the infested leaves at 6 hours. But thereafter, the response is smaller in terms of the number of transcripts changed. Within the local leaves, you see the usual suspects in terms of the components which are responding. So these are salicylic acid associated transcripts that you see here, with marker transcripts for salicylic acid, marker transcripts for ethylene associated changes, and marker transcripts for abscisic acid associated changes. But what is perhaps more interesting is the divergent response between the local infested leaves and the systemic leaves. So within the local response, you can look at the types of transcripts that are changed and compare them to the systemic leaves. And you see here that there are quite divergence. There is a divergence in the types of transcripts changed. For example, there are RNA processing transcripts which are very much changed in the local leaves. While some are still changed in infested leaves, there are other components such as lipid metabolism which are much more strongly changed in the systemic response. So when we'd finished our profiling, we compared our profiles to all of the other transcriptome profiles in the literature. And actually, there were very few constant changes. But what was particularly important, and is not often perhaps mentioned in plant responses to aphids, is the importance of the cell wall response. And in all of the transcriptomes that we looked at, you can see here some of the components that were changed, but in relation to cell wall and cell wall associated processes. And I will come back to that later in my talk. So I'm particularly interested in redox responses, and our transcriptome signatures revealed how strong the redox response is. So there is a burst of reactive oxygen upon perception of the aphids. And here, the most strongly upregulated transcript within 6 hours is redox responsive transcription factor 1, which is an important transcription factor in the amplification of the redox signal. Also, the second is one of the heat shock factors here, which is, as you see from this figure below, induced in response to many stresses. But we decided to move forward in the early days in looking at the importance of the role of oxidative and reductive processes in the plant response to aphids. Now, aphids are supposed to be stealthy feeders. They don't cause as much damage as other insects. They have a probing proboscis that penetrates between the leaf cells. However, the aphid actually tastes every cell on the way down, looking for the components, the sucrose and the amino acids that are in the phloem. So every cell on the way down is damaged. And you can see a burst of reactive oxygen that follows the path of the proboscis. It goes down to the phloem cells. That burst of reactive oxygen is accompanied by signaling within the cell, signaling on the cell membrane, and signaling within the cell, which is redox signaling and related signaling. Now, this looks a complex slide, but in essence, all it says is the cell is receptive to both oxidative signals and to reductive capacity, which limits the lifetime of those signals. So we have oxidants and antioxidant components. And I just want to show you how important those antioxidant responses are and the relationship between oxidants and antioxidants. So a simple experiment. You can take leaves and feed them with the precursor of ascorbic acid. So ascorbic acid, vitamin C, is a very important antioxidant in plants. If you take the precursor and feed it to leaves, then in this slide, you can see there is a gradual rise in the ascorbic acid content. And as you increase the amount of the antioxidant, the survival of the aphids, as you see here below, is also increased. And that occurs despite the fact that the actual amount of ascorbate, which is in the aphid, remains relatively constant. Most of it is excreted in the honey tube. So it suggests that the ascorbate is important in terms of the plant response. And we were able to show that very nicely by looking at mutants which are deficient in ascorbate synthesis. So here is a wild type Arabidopsis leaf and a mutant which is lacking ascorbic acid, vitamin C, ascorbic acid, so it's VTC2. And you can see that the reproduction of the aphid on the leaves with low ascorbate is significantly lower. Now, that is related to signaling which is downstream. In this case, we've shown that it's related to abscisic acid signaling, because we can take mutants which are lacking in abscisic acid signaling and cross them with our VTC2 mutant here, the ABI4 VTC2. And you can see the aphid fecundity in the double mutant ABI4, the mutant that lacks abscisic acid signaling as well as low ascorbate, remains the same as the wild type despite low ascorbate. So the ascorbate itself is buffering the signaling capacity, which leads to downstream signaling through phytohormones and other processes. Another important feature is the wall, as I said to you before, and we can look at the wall from a redox point of view. One of the important components of the wall is ascorbate oxidase. So ascorbate oxidase is an enzyme that lives in the apoplast and the cell wall compartment. So you have bursts of reactive oxygen in the apoplast, which are due to the presence of the aphid. But the redox balance in the apoplast can be changed by manipulating ascorbate oxidase, which is solely located in the wall. And we've done those experiments in tobacco. And if you look at the top panel here, you can see ascorbate oxidase activity. The wild type's in the middle. And on one side, we've got what we call PAO plants, which have lots of ascorbate oxidase, and then TAO plants, where we've manipulated them to have low ascorbate oxidase. The black bars show the effect of aphids. So the plant response to aphids is immediately to lower the ascorbate oxidase content. But when you look at aphid fecundity, the bottom panel, in relation to the amount of ascorbate oxidase, you can see that the more ascorbate oxidase there is in the plant, the more aphids are able to reproduce. So the ascorbate content of the wall, the ability to battle reactive oxygen in the wall is really, really important, so that the plants with the low ascorbate oxidase are able to limit the infestation better. And I won't show you a lot of the detail, but a lot of that is to do with regulation of the structure of the wall. And here, this is a complex slide, but you don't need to look at much of it, but you can measure all of the carbohydrate components of the wall using antibodies. And this is literally what we've done here. If you look at the middle sections C and D here, and you look at the qualitative differences. So for example, here in this brown peak here, in the absence of aphids, it's very high. In the presence of aphids, it goes down considerably. And we can see that in the plants with the low ascorbate oxidase, that change is much reduced. And here are some of the details here. If you want to look in more details, please go and look at the paper. And you can see also in terms of the expression of genes encoding proteins involved in the wall B here, that there is a big change which is manipulated by the aphids and is part of the response to the aphids. You can, I've shown you data with ascorbic acid. You can do it with many other of the antioxidants. Here's data with CAT2. I'm not going to go into the detail here, because the upshot is that when companies are looking for an easy way perhaps to manipulate aphid resistance, the antioxidant genes are large families, and it's possible to look for variants with lower antioxidant activity that wouldn't affect abiotic stress tolerance, but would increase the resistance to aphids. Now, if I've got, shall I finish here, or have I got 2 more minutes to carry on?
You've got a couple more minutes, Christine.
Thank you. So this is what we're doing now. So I showed you some of the usual suspect hormones in relation to resistance to aphids. But plant has many hormones which are involved in plant processes. And part of our transcriptome profile suggested hormones called strigolactones were also important to responses to aphids. These hormones control branching, they control symbiotic interactions with other organisms. And it's been increasingly realized that they're involved in plant defenses. For example, there was a recent paper on nematodes. So we've been looking at the effect of strigolactones. We've chosen to do this work in P because P aphids are very important. So what we can do is look at the role of strigolactones by taking mutants which are either defected in synthesis or signaling of strigolactones. We grow them both in air and ICO2 because combined stress is interesting as well as the single stress. But you can see the effect of loss of strigolactones here. The plants are much more branched. So branching, you can see the control of branching by in these mutants. So we take the P aphid, which is a really important aphid, and we look at the reproduction of the P aphid in the wild type and the mutants. And you can see here, I think, the aphid fecundity is actually reduced in mutants which lack strigalactone synthesis and signaling quite significantly. So we are now exploring this, and we can look at all of the other hormones in these mutants. We do it in the same way that we do the fecundity test. So we're only doing with a small amount of aphids. But one of the interesting features is here that you can see there's a strong effect of on the gibberellic acid content of the leaves, both by high CO2 and by aphid feeding. And there is, in fact, a relationship between gibberellic acid, strigalactones, and aphid fecundity, which we will explore further. So I'm going to finish there and thank the people who've worked both in my team and my collaborators. But I hope that I've shown you that there are many components that are involved in the plant response to aphids. It is possible to limit aphid fecundity by manipulating those components. But we still have to work in a real world context where we see the aphids not on their own, but looking at the combination of other stresses, the abiotic stresses, heat and drought, as well as aphid feeding. Thank you for listening.
Thanks very much, Christine. And we've got a couple of questions for you in the chat. The first question is from Graham Kettles. Graham, would you like to ask your question?
Yeah, sure. Thanks, Nicola. Thanks very much, Christine. Yeah, I was wondering if you have done similar experiments with any species of aphids that you could consider as true specialists of brassicas or Arabidopsis, and whether you see the same systemic transcriptional responses, or whether they're better able to prevent that process, that activation of defense from happening.
Yeah, I've never worked with a specialist feeder of Arabidopsis. That would be an interesting thing to do. Within our transcriptome profiling, though, there were clear differences in the responses of Arabidopsis to generalist feeders and to specific feeders, but they were largely within the scope of secondary compound production. It was quite interesting. So the hormone responses were quite similar, but we found that within the transcriptome profiles, the specialist feeders seem to induce secondary compounds much more than the generalist feeders. But that's all I know.
Great, thanks very much.
Thanks, Christine. And then we've also got a question from Florian Bush. Florian, go ahead.
Yeah, thanks for the talk. In one of the earlier slides, you showed that the aphid survival on systemic leaves after infection is lower than the survival of the actually infected leaves.
Yeah.
Which would indicate or my question would be, do the locally infected leaves not benefit from that systemic response in the same way? Or is there anything in those infected leaves that actually prevents a full systemic response?
The systemic, I think the answer is to do with the induction of programmed cell death and damage within the local response. So when we do these experiments, we tend to put 50 aphids within a clip cage. Even though we measure 6 to 8 hours, as the first time point, I think there are local damage responses within the local leaf that make them less able to, they can withstand the infestation better than naive leaves that have never seen an infestation. But the systemic leaves seem to fare better. And my view at the moment is that they don't go into those programmed cell death type pathways that heavy infestation causes on the local leaf.
Great, thanks.
Thanks, Christine. I just had a question about, is there any evidence